Okay, so this should be the second video to complete your first note packet for our cell cycle set of notes. And so what we're going to be looking at here are the actual phases and details about the phases of the cell cycle. And so we're going to go through interphase and mitosis in this presentation. We're first going to break down interphase, which starts with the formation of a cell and ends right as the cell enters mitosis. This is going to be the main life stage of each cell, and as we see, or as you're going to see, it's divided into three distinct parts, G1, S, and G2. Okay. Interphase then is the time the cell is going under normal growth. It copies its DNA and it prepares to divide. And so each of these things, 1, 2, 3, fit right in with the growth phase, the copying phase, and the preparation phase. Now, as you learned in the POGIL, those are actually called the G1 or GAP1, S, and G2 or GAP2 phases. I use the words that are over here, growing, copying, and preparing, to help you to remember and distinguish between the phases. So the growth phase is cells growing to mature size, going from that small cell that forms from mitosis to the overall larger size for that cell type. The copying phase is when the cell's making a copy of its DNA, or S you can remember for synthesis, copying the DNA, synthesizing new DNA. And then G2 is a very short but rapid period of growth, preparing, so trying to swell the cell very quickly in order to divide. And so that, what we just talked about, that's occurring in this whole range here, okay? So all of this is considered to be interphase. And so a cell spends most of its life, most of a cell's life in the interphase part of the cycle. Okay. This would be just a picture of a typical cell found in interphase. I want to highlight so you can see the membrane out here. Okay. Here's the nucleus in the center. Notice you don't see any chromosomes. The DNA is not packaged tightly or wound up. And so this is just, it looks like a normal cell. And it would be hard to distinguish on the microscope whether it's in, for instance, G1, S, or G2. The cell is then, when it's ready, after it goes through the complete G2 phase, it's going to enter what's called mitosis. So this is the process by which cells divide their nucleus. It's only their nucleus, all right? So mitosis has 4 subparts that we're going to break down, but this is what you refer to as cell division. Mitosis starts with prophase. Now, I like to match up the first letter of each phase with a word to help to remind you what's happening. And so prophase is when the cell's preparing to divide. And so basically during this phase, the cell takes its chromatin, which is, remember, the loose DNA, and it condenses it into chromosomes. So it takes the loose, plate of spaghetti, and then it winds it up into that condensed X-shaped structure, the chromosome. During prophase, the nuclear membrane disappears, okay, and your centrioles, which are your anchor points, move towards opposite sides of the cell, and your spindle fibers form, okay? And so we're going to look at a picture. I'm going to highlight some of these structures. All right, so check it out. Here's where our nuclear membrane was, and you can see, like right here and over here, there are now gaps in the membrane because the nucleus is dissolving. As a reminder, all the way out here is our cell membrane, okay? Then when we look a little bit more closely, you're starting to see these like dark structures, some of which are in an X. These are the chromosomes that are forming up. And in here, it's hard to see, but you have the centrioles forming. And so the centrioles and the spindle fibers would be like over in this region at the moment. So nucleus dissolves, step 1. Chromosomes coil, step 2. Spindle fibers and centrioles move out, part 3. The next phase is going to be metaphase, okay? And I would say metaphase is in the middle, okay? And so the key thing here is the chromosomes actually move. So if this is your cell, the chromosomes actually move and line up in a straight line down the middle of each cell, okay? And so our word here is middle. Basically, how that happens is each chromosome is attached to a spindle fiber, and the spindle fibers are attached to the centrioles, and they push and pull the chromosomes. So, they can move them left, right, whatever, to get them in a line down the middle. So we're going to look at that. So here's a cell in metaphase, and this is great. You can really see the chromosomes are lined up here right down the middle, right? The centrioles are right here in the green, and the spindle fibers are these linear things that I'm going to highlight with blue. Okay. Notice at this point we have no nucleus, and once again this is metaphase or middle. And so this diagram gives a general overview of what we looked at so far. And so we've got interphase, prophase, and metaphase. So in between, preparing, and then middle. And so down here are the key sort of features of each phase. Up next is anaphase. I like to say that anaphase is when they move apart. And so what happens here is, remember, we have chromatid. So chromatid is essentially, it's 1/2 of the chromosome. Let me do that in chromosomes in red. So chromatid is 1/2 of the chromosome, and they get pulled apart, right? And so you have your X, and what happens is the spindle fibers pull them away from each other or apart, and so they get moved towards opposite ends of the cell. And so what's going to happen is you need to remember that each side is a complete copy, and so you're moving one complete copy of DNA towards either end of the cell. So here's that again. So we've got, remember, our centrioles over here. Our spindle fibers are actually here. They're contracting. And now, instead of X's, we have 1/2 of the X of the chromosomes moving in either direction. Notice still no nucleus. Cell membrane is still out here. And so we're just watching as this cell is dividing its nucleus. Our next phase is telophase, and I like to say telophase is 2, and what we have 2 of here by the end of telophase is 2 nuclei. What happens here is that the chromosomes move towards opposite ends of the cell. And so remember what happened is they were split, right? They moved away from each other or apart, and now you've got a cluster of chromatins at each side of the cell. We now call them chromosomes again. Basically 2 new nucleuses start to form around each cluster of chromosomes, and so we end up with 2 nuclei. So you've got 1 cell membrane but 2 nuclei. I'm going to show you a picture of this in just 1 second. Now, a lot of books, and the picture's coming up, but a lot of books and different teachers will lump this cycle, cytokinesis, as part of telophase. We're going to look at it as a separate process, and they're going to be distinct from each other. And so cytokinesis, look at that word. Cyto means cell, okay? And so during cytokinesis, the membrane of that cell pinches in at the middle, and the original cell splits into 2. And so that looks basically like you've got a membrane, it's pinching in the middle, right here down a line, and then it turns into 2 distinct cells. So what we're looking at here, and this is the reason why a lot of books will lump cytokinesis and telophase into one process, is that the cells don't like pause in the middle and say, okay, I'm going to enter a new phase. And so this cell right here is actually, it's doing both things at one time. And so here's the chromatids, or the chromosomes, after they've been separated, clustered in each cell. Notice the membranes are already starting to pinch in. And if you look closely, these condensed chromosomes are starting to form into new nuclei. And so, well, which is which? The process of forming the new nuclei is telophase. The process of pinching the membranes in is cytokinesis. So I think this picture is really neat. This is an electron microscope picture, color added, of one bacterial cell that's going through cytokinesis. It's at the very end of pinching off to form 2 new cells. And so, just to show you that this is happening in real cell types, and is it's an interesting process. So this diagram should help you study, and it's just breaking down the final couple of phases. So we've got anaphase, which, remember, is apart. The chromatids are being pulled apart. We've got telophase, that's 2 nuclei, 1 and 2, and then cytokinesis, that's the pinching in of the membranes here. And then where are we in the cell cycle after that? Well, we're right back to where we started. So as soon as cytokinesis is over, we're right back in interphase, and that cell is just starting the cycle all over again. Now, there's just a couple of distinctions to make between plant cells and animal or protocells going through this process. The main thing is that plant cells, they've got, right, a cell wall. So what are they doing with the cell wall? Plants actually make a whole extra piece during division, and it's after anaphase. They start to line up some vesicles along the middle of their cell, and they form a structure called the cell plate. So this is the cell plate. The plate is basically the start of a new cell wall. Okay. The main other difference is that plant cells don't have centrioles. They use their cell walls as anchor points for their spindle fibers, which should make sense because they're a rigid structure that they can push and pull against. So the cell plate, remember, is the new cell walls as they form. Just to review, we've got, we're starting at inter and then pro, meta, ana, telophases. These on their own make up the M phase of mitosis, separate from interphase. And then just to revisit the whole process, we've got the formation of the new cell. Okay, it then travels through G1, that's growth. S, this is synthesis, copying the DNA. G2, this is rapid growth and preparation and division. We then have a very short period of time for interphase where we do prophase, metaphase, anaphase, telophase, and wrap it up with cytokinesis, which is taking that one cell that has 2 copies of DNA and turning it into, eventually, 2 cells that each have 1 copy of the DNA that are going right back into interphase again.